طيب بسم الله الرحمن الرحيم. اليوم حنحكي عن السكند ليكتشر اللي هو حنكمل فيه ال innate وال adaptive immune system. في ال last lecture حكينا عن ال two main divisions of the immune system. حكينا عن ال innate وحكينا عن ال adaptive. هاي الصفحة واللي بعديها حتكون مراجعة. طبعا حتلاحظوا انه في التفريغ هذا فيه اشياء مكررة كثير وتم التعديل عليها علميا، يعني تعديلات كثير ما قدرنا نحصيها. لو قدرنا نحصيها كان حطيناها في correction zone والله المستعان. طيب ال innate that we are born with، هسه زي ما بنعرف انه ال innate the first line of defense، اول خط للحماية في الجسم. هو non-specific so the innate immune system deals with any microorganisms or any infection لأنه ما عنده specificity، وبالتالي he deals with microorganisms مش specific and no memory that is involved. هسا هو ال first line of defense وال second line. ال first line of defense هو عبارة عن ال innate وال second line of defense عبارة عن innate. ال first line of defense بنحكي عن بنسميهم ال physical barriers اللي هم ال skin و mucous membrane. وبنعرف انه ال skin اول خط دفاع لنا في الجسم. وايش الميوكس ممبرين؟ الميوكس ممبرين احنا بنعرف انه اي ميوكس ممبرين that lines a cavity. هسا الكافيتي هاي محمية بميوكس ممبرين متى؟ لما يكون عرضة لايش؟ للاكسترنال environment، لأن الاكسترنال environment هو اللي فيه الانفكشنز واللي فيه المايكرو اورجانيزمز. هسا let's take an example about the mucous membrane. هسا ال respiratory system فيه mucous membrane، تمام؟ وفيه ايش؟ sticky mucus، اوكي؟ وفي سيليا. هذا الاشي بيمكننا انه when the infection or microorganisms or viruses or bacteria go through this path or this tract, it will get stuck in the sticky mucus. طيب عندنا كمان الاسيد في ال digestive system. احنا بنعرف قد بناكل ايش؟ بناكل اكل كثير، فيمكن يحتوي الأكل هذا على infections، على مرض، على pathogens، على أي إشي مضر، وبالتالي أن الأسيد اللي هو بوصل ال pH تبعه 2 وبالتالي بقتل أي microorganisms as well as the saliva in our mouth. تمام. Now sometimes the microorganisms like bacteria damage and pass through the physical barriers. تمام. هسا we will get infection. هسا عندنا حالتين، dehydration اللي هو first cause of death and burns which is the second cause of death. هسا both of them will cause what? losing the physical barriers، the skin أو mucous membrane أو mucous membrane. هسا if we lose one of the physical barriers, so we lost what? The first line of defense which is the skin and mucous membrane، وبالتالي حيؤدي إلى إيش؟ عرضة للإنفكشن أكثر. طيب we talked also about sebaceous glands and sebum that produce fatty acids، and حكينا عن الصوديوم chloride. الثلاثة دول inhibits the growth of the microorganisms, so we need it. Now let's talk about the second line of defense. The second line of defense are composed of group of phagocytic cells, and the most common two are the neutrophils and the macrophages. And the neutrophils, which is the largest cell in number among white blood cells. Now, but the problem, neutrophils have a short half-life. And why is that? Because its mechanism of killing bacteria or infection is makes it dies immediately. Now, if we have an infection, the neutrophils will go to the site of infection and will deal with what? Will attack the foreign body and deal with it. After that, it will phagocytose it. okay? When it phagocytoses it, it will die. And when it die ايش حيظهر عنا؟ البus. Okay, macrophages. Macrophages, they are larger and they are less in number, but they do not expire by the act or infection. So they live, which means they stay in our body for a longer period of time. Now, we have something called antigen presenting cell. من الاسم, there are a group of cells that present the antigen to a specific type of cells. Okay? There are different type of cells like macrophages and dendritic cells that help the immune system by presenting the antigen to T and B, mainly to T helper cells, for a specific immune response. Okay? And as we will see that macrophages help the T helper cell to recognize a specific antigen by presenting it to the T helper cell. يعني أنا التي هيلب ال APC مثلا لو ناخذ الماكروفيجز على سبيل المثال، الماكروفيجز مثلا اللهم صل على محمد، في شيء معين، في antigen معين T helper cells cannot recognize. So what will happen? The macrophages will present هيقدم هذا الانتيجين for the T helper cell. Okay, next هستخدمها. Okay, next الانتيجين هيجي يرتبط معه إشي إيش؟ اسمه MHC، والMHC اسمه اللي هو Major Histocompatibility Complex. هسا هذا MHC هيرتبط مع إيش؟ الانتيجين and this attachment will cause an activation in what? T helper cells. لما يصير activation في T helper cell, it will produce cytokines. Okay? And why do we need cytokines? To continue the specific immune response. So خلينا نلخصها أن ال APC are different type of cells like macrophages, dendritic cells that help the immune system by presenting the antigen to the T helper cells or T and B cells. بعدين بيجي ال MHC complex بيرتبط مع الانتيجين. فلما يرتبط مع الانتيجين the T helper cells will activate and produce what? Cytokines that are needed to continue a specific immune response. Now, what will happen if what will happen? Now, let's talk about the adaptive immune system, which is the third line of defense. Now, there's a specificity where an epitope, which is an antigenic determinant. And what is the antigenic determinant? It is a part of the antigen. It is a specific part of the antigen. هذا إيش؟ اسمه الانتيجينيك determinant هو الepitope. So this epitope, which is a part of the antigen, must hook into a receptor. تمام؟ بيرتبطوا مع بعض like lock and key. Then epitope, which is the antigenic determinant, which is a specific part of the antigen, will hook and attach to the receptor, which is the نسميه paratope, like a lock and key. Now, T cell receptors, as well as antibodies, give the specificity for the adaptive immune response. Now, the adaptive immunity is activated by what? Exposure to pathogens, and uses an immunological memory to learn about the threat and enhance the immune response accordingly. Now, هسه الإشي العبقري في الادابتيف immune system هسه لما يفشلوا the first two lines of defense هنيجي على الادابتيف he is much slower. تمام؟ الادابتيف much slower بس عنده شغلة ميزة اللي هي memory cells. The memory cells, they recognize the antigen if they are exposed to it in the second time, and they don't just recognize it, they recognize and know how to deal with it. Subhanallah. Now, B and T cells receptors are already performed based on our genetics. So we are born with a set of receptors that are ready to bind to and get rid of the invading microorganisms. The adaptive immune system is slow because the antigen takes time to bind with the receptor. قلنا الابيتوب مع الباراتوب لوك اند كي. هسه بصير عندنا شيء اسمه clonal activation. هسه the clonal activation, products formation and cytokines release is slow. هاي في الادابتيف immune system. واللي في الكلونال activation ايش يعني clonal activation؟ بصير انا اعمل ايش؟ cloning, duplication, okay, in the number of lymphocytes or cells. Why؟ أنا كل ما أزيد السيلز كل ما أزيد الريسبتورز على السيلز. كل ما بتزداد الريسبتور حينزاد إيش؟ ارتباطهم بالانتيجنز. طيب. However, the adaptive response will be faster in the next exposure for the same antigen because we will have memory cells. Adaptive recognizes what self from non-self antigens in the primary lymphoid organs. In the primary lymphoid organs, يعني we're talking about the thymus and the bone marrow. طيب. طيب هون هنعيد اللي حكيناه. هسا the essential features of the host defense system comprise an innate component that functions as the first line of defense, and an adaptive that takes longer to mobilize but confers specificity and memory cells. As shown in the overview figure above, the two compartments are not independent, okay, but are functionally interrelated between each other. So, in a critical ways, such as the cytokines through the actions of soluble effector molecules called cytokines. Now, the cytokines are secreted by the APC, antigen-presenting cells. These cytokines are soluble effector molecules or hormones that are secreted from certain type of cells and what to affect other type of cells to produce a specific things. Like cytokines is secreted to see that cytokines will activate B cells and to produce plasma cells and antibodies. Now, what is the antigen presenting cells? Again, antigen presenting cell like macrophages, dendritic cells, or even B cells have to present the antigen to the T and B cells to produce adaptive response. Now, what is the complement system? Complement system. Now, the complement system in serum and how it functions non-specifically to lyse cells before the presence of inflammatory cells in area by chemotaxis. Now, what will happen? Now, this complement system, if there's an inflammation, okay, it will come and start lysing cells before the presence of inflammatory cells. Now, after lysing the cells, it will call the neutrophils by what? Chemotaxis. The chemotaxis is what? Is a messenger molecule or that signals the inflammatory cells to come to the site of the inflammation. So this complement system works through a cascade by destroying cells and then call the inflammatory cells to cause inflammation. The complement system provides an innate immune response. This is why. Okay. Now, adaptive immune system, third line of defense, as we know, is mediated by T cells, the helper 1 or 2 or even 17. Now, each T helper cells produces different types of cytokines for different functions. And T helper cells produces another thing, which is T cytotoxic. the T lymphocytes will activate and B cells that differentiate into plasma cells. And then the plasma cells or antibodies will secrete immunoglobulin that will fight the infection. This process will be discussed in further lecture. Now, let's focus on the figure in here. Now, the figure above has two yellow or orange represents the infection, and the green represents the innate immune system. This is the innate immune system. Okay, and the blue represent what? The adaptive immune system. Now, if there is an infection, viral infection or bacterial infection, the first line of defense will be the innate immune system through the complement system, as we mentioned, and the antigen presenting cells like macrophages, neutrophils. Now, the activated macrophages will present what? The antigen to T cells. Present the antigen to T cells. And produce what? Cytokines. Now, the production of cytokines will what? Turn and activate the T lymphocytes. So ذكرنا ما قلنا في ارتباط بينهم كيف عن طريق السيتوكاينز هيك السيتوكاينز. إذا cytokines, after the macrophages is activated, it will secrete what? It will secrete cytokines which will activate the T lymphocytes. Now, if the first and second line of defense fail to prevent the infection, signals will be sent to the third line, which is the adaptive immune system, cell-mediated immunity humor. هسا عندنا three types of antigen recognition molecules are used by the immune system. Antigen recognition molecules, ARM. Now, we have B and T cells receptors. We have B cell receptors and T cells receptors, and we have the major histocompatibility complex. هسا ايش يعني antigen recognition molecule؟ Molecules that recognize the antigen. Okay, B and T cells receptors. Now, both B and T cell receptors have the same protein structure on the surfaces, which is the immunoglobulin folds, and it is هاي معلومة هيك which is a domain consists of what? A pair of beta sheets linked by a disulfide bond and hydrophobic interactions. Now, that's B and C B cell receptors. The B cell lymphocytes, as we know, are responsible for a humoral adaptive response, which involves the production of highly specific antibodies. The B or T cell is specific for a particular antigen, but they are different in how they detect those antigen, how they detect, how they recognize كيف يميزوا هذا الانتيجين. هسه T cell receptors once formed in when? Once formed in the bone marrow, T progenitor cells migrate to thymus to mature and become T cells. While in thymus, the developing T cells starts to express what? T cell receptors, TCR, and other receptors called CD4, CD8 receptors. هسه كل T cells express T cell receptors, either CD4 or CD8, not both. يعني واحد منهم. هسه unlike antibodies اللي حكينا عنه في ال B receptors. هسه antibodies can bind to the antigen directly, but T cell receptors can only recognize antigen that are bound to certain receptor molecules. Mean certain receptor molecules that we talked about them هم called major histocompatibility complex MHC. قبل شوي حكينا وين أعتقد حكينا الأنتيجين major histocompatibility will what attach to each other. Now نلاحظ احنا قاعدين نعيد. اوكي هسه هذول major histocompatibility complex في عندنا two classes, class 1 MHC1 and MHC2. These MHC molecules are membrane bound receptors on the antigen presenting cells like dendritic cells and macrophages. هسه CD4 and CD8 play a role in T cell recognition and activation by binding to either MHC1 or MHC2. Now, what is the difference between the BCR and TCR? Yani B cell receptors and T cell receptors. Now, the receptors on the surface of B cells can bind to soluble antigens directly, while T cell receptors can only recognize an antigen which is complexed with the major histocompatibility molecules on the surface of the other cells. هنعيد هاي المعلومة آخر شيء. Okay, what are the MHC? MHC هم antigen recognition molecules. Well, MHC1 found in all nucleated cells. MHC2 found MHC found in professional antigen presenting cells like macrophages, and B cells have MHC class 2 proteins. Now both functions in regulating natural killer 2 and present the antigen to T cells. هسه نحكي بسرعة عن ال innate immune system. حكينا إنه ال innate immune system is fast and unlike the adaptive system which may take days to mobilize. هاي احنا نحكي على adaptive. Now the aspects of the innate system are extremely quickly mobilized. هسه احنا قلنا انه innate كمان macrophages بتدخل فيها. For example, phagocytic cells like macrophages resident in tissue will recognize infection via pattern of recognition molecules that detect structural motifs on invading microbes. إذا innate immune system في عنا the second line of defense which is the phagocytic cells, which is the macrophages. As these macrophages, how they detect or recognize the infection via a pattern of recognition molecules. What are the recognition molecules? We have a toll-like receptors and we have the Manon binding lectin. إيش معنى؟ إنه they recognize okay the antigen بعلامة مميزة. يعني أنا شفت أنتيجين عليه شوجر من فوق فبعرف إنه هذا كذا كذا فبحاربه. هيك المبدأ يعني. Now toll-like receptors TLRS toll-like receptors recognize a variety of substances found on a range of microbes but not on host cells. هذا Manon binding lectin of the complement system. Now this Manon binding lectin which recognizes sugar molecules containing mannose on the surface of bacteria, fungi, and viruses and helps activate the complement cascade. MBL is present in solution in plasma. So when they recognize a sugar mannose on the surface of bacteria or fungi and viruses, they will activate a complement cascade. So this how innate immune system deals with microbes or microorganisms. They recognize them by a pattern of specific recognition molecules like toll-like receptors and the Manon binding lectin. The innate system uses phagocytic cells, chiefly neutrophils and macrophages, in addition to molecules such as the serum proteins of the complement system, which can interact directly with the certain microbes to protect the host and lyse them. The other cells important in the innate response are the NK cells, which can detect and kill certain virally infected cells by inducing programmed cell death, apoptosis. Another group of important soluble molecules that is a part of the innate defense system is the interferons. And the interferons, as we know, is produced by the infected cells. Interferons inhibits the replication of many viruses and is not pathogen specific. But now, many Many innate system components, for example complement, interferon, cytokines or cells such as macrophages can affect cells of the specific adaptive system وهذا بيبين لنا ايش قديش ترابط بينهم. This is another important observation. The innate and adaptive systems are interconnected and overlapping. The adaptive system is usually triggered by the innate system, and it only comes into play if the innate system fails to overwhelm the invading microbes or if the invading microbe has found a way to avoid interaction with the innate system. طيب هون إشي بسيط cellular and molecular components of the immune system. Cellular and molecular components of the immune system. Now, neutrophils, okay, when we have an inflammation or trauma, chemotactic factors will be secreted to call what? The neutrophils, okay, immune cells. Now, هسه we see neutrophils accounts تقريبا 50 to 70 of white blood cells, and it is used to kill bacterial infections. So when you read an CBC and report and find these cells with a huge number, that means there is an bacterial infection or inflammation. As an eosinophil, we can see this type of cell if there is a parasitic infection like worms and protozoa, or in allergies, type 1 hypersensitivity. They are normally about 5 of white blood cells, but they can reach 20 to 30 in the parasitic infection. Now, macrophages, they are about 5 to 10 phagocytic cells and play a role in antigen presentation. Have granules, they do not expire by the act. حكينا عنهم. They are important in cell-mediated immunity in chronic type infection like microbacteria. Natural killer, detect and kill cancer cells and certain virally infected cells by inducing programmed cell death apoptosis. So it kills our own cells by apoptosis. Now, dendritic cells, our infected cells عشان نعمل الانتشار. Dendritic cells, antigen presenting cells. Looks like macrophages, but the main function is to present antigen through class 2 MHC antigen to helper cells. Through عفوا هاي through. We have mast cells, okay, have some inflammatory mediators stored in granules and receptors of immunoglobulin E antibodies. So عنده stored granules زي الهستامين, leukotriene. They are inflammatory mediators, and mast cells are receptors of IgE antibodies, immunoglobulin antibodies. هسه الانترفيرونز قلنا inhibit the replication of many viruses and cancer cells. وقلنا the complement system can interact directly with certain microbes to protect the host cells before the inflammatory cells come to the place of infection. Also, it facilitates phagocytosis. مين هذه complement system. Plasma cells, they are formed from B cells and ready to produce antibody. How they are formed? We knew that the cytokines are produced by B cells that will make plasma cells antibodies. هنيجي نحكي. Now, how can we differentiate between T helper and cytotoxic cells from the receptor? T helper cells have CD4 receptor, okay, but T cytotoxic have CD8, okay, I CD8 and CD4, okay, the adaptive immune system. Like adaptive immune system is capable of distinguishing self from self. It is specific, and this specificity is accomplished by creating what? Anti-capillary defense system of diverse recognition molecules, meaning that we will duplicate ourselves and to have many receptors that will interact with many foreign bodies and antigens. Now, the adaptive immune system develop memory cells, so we can remember the antigen in the second exposure, and we can be more efficient to deal with it. Vertebrate genomes contain several genes that encode many millions antigen recognition molecules. These gene families include antigen receptors capable of recognizing any given antigen. As in this figure, a molecular mechanism enables some of the receptors, human antibodies, okay, some of the receptors or antibodies at the somatic level during the immune response to create what? Receptors with a better fit. That is, the binding is more specific. Fit to what? Fit to the antigen. Now, this ability to generate anti-capillary defense system against non-self entities was enhanced by what? Duplication of those genes that encode proteins that have binding sites and code function as receptors. Now, the products of these genes duplication located in the short arm of the chromosome number 6. Now, as the gene families that encode the antigen recognition molecules حكينا عنهم T cell receptors and major histocompatibility. Okay? Okay. Now, each lymphocyte expresses a unique antigen receptor. Once an antigen encounters a lymphocyte bearing the receptor that best fits the antigen, this pre-existing cell divides and gives rise to many daughter cells, clones. هون هاي مفروض نحكي عن هاي, okay? Now, notice in the figure how the innate has no major change genetically, but which makes it low specificity and diversity, innate. But regarding the adaptive immune system, the genetic changes are extremely important. Okay. Now, in this figure, each B lymphocyte have a specific receptor for a specific what? Antigen. When the antigen comes and binds, the lymphocytes start to proliferate, duplicate, cloning, زيد عددها, زيد the receptors. One complication of an anti-capillary system with pre-existing receptors is the generation of anti-self receptors. As the anti-self receptors in a healthy person have to be inactivated or deleted. When mistakes occur and potentially anti-self cells are allowed to remain active, so autoimmune disease can occur. So any mistake in the anti-self cell activation or deactivation will cause autoimmune disease if it is activated. طيب هذول كل هاي الصفحة تقريبا من هون, كل هذول من الكتاب, تمام. Now, let's talk about the components of the immune system. Specificity طبعا, diversity, and number cells. حكينا عن cells and soluble molecules. حكينا عن all cells and their function. حكينا عن cell-mediated immunity involves T-cytotoxic and T helper cells, and the humoral immune response involves antibodies and T2 helper cells. Antigen presenting cells and function. طيب هذول components of the immune system. طيب remember, cell-mediated immunity, cells involved in destruction of other cells, virally infected cells or tumor cells. Humoral immune response, antibody-mediated. The antibodies will neutralize the virus. They will bind to the virus and prevent the virus from binding to its receptor. هذا الhumoral immune response. APS, macrophages, and dendritic cells and B cells are APCs. They have the class 2 of MHC, so they can present the antigen to T helper cells, so the antigen must fit MHC class, the receptor, in something called the restriction phenomenon. This is a restriction phenomenon. Okay, so what is the restriction phenomenon? That macrophages or dendritic cells and B cells, okay, have a class of MHC2 so they can present the antigen to T helper cells. حكينا. So the antigen must fit MHC class 2. This is called the restriction phenomena, and we talked about it in the previous pages. Now, phases of an immune response. First phase, an الconjugative phase. And what will happen in this phase, which is the antigen enters our body and select the antigen with its specific receptor on the B cells. So this is a B cells, and this is a receptor or a specific receptor for a specific antigen that will bind to it. And secondly, activation phase. What will happen? Clonal expansion and activation proliferation, making more matchmaking receptors. The clones will be activated, changed to plasma cells. Okay, why we need plasma cells? To produce antibodies and to use it in the effector phase in antigen elimination. Okay, طيب هسا عندنا some antigens can stimulate the immune response directly على طول. أول ما يدخل الأنتيجين stimulates the immune response على طول. T cell dependent, okay, acquired immune responses typically require antigen presenting cells to present antigen derived peptides within major histocompatibility complex molecules. هاي شرحناها قبل شوي. حكينا إنه في إشي اسمه T cell dependent، حنحكي بعد شوي. T cell dependent و T cell independent. T independent response و T dependent response. ال T independent response حكينا حنحكي إنه حنشوف إنه in T dependent response the antigen is ingested and attached to MHC molecule, so we need the MHC molecule to recognize the antigen. بينما في ال T independent response the B cell will bind directly to the antigen. So the antigen presenting cells are macrophages, dendritic cells, and B cells. طيب. طيب. Types of immune response. هسه the components of the immune system makes 3 different types of immune response depending on the habits of the invading pathogen. Whether it is intracellular pathogens or extracellular pathogens or surface pathogens. Okay, so على أساس type of the pathogen, I take a specific type of immune response. Now, this table summarizes these types which can cause very common diseases. Like مثلا ناخذ كل واحد مثال على السريع. Intracellular pathogens عندنا myobacteria. هاي طبعا myobacteria example tuberculosis, viruses, protozoa. Type the innate immune system components and the interferons and macrophages, natural killer cells, and adaptive immune system components, Th1 T cells, cytotoxic T, lymphocytes, immunoglobulin M, G, and A, immunopathology, autoimmune disease. ايضا هون autoimmune disease but surface pathogens حتسبب لي ايش؟ allergy. وهكذا. هذا الجدول حفظ الله يعطيكم العافيه. Now we have something called the active and passive. We have active and passive immunity. The active immunity occurs when the individual plays a direct role in responding to the antigen. الباسيف هو the immunity is transferred from one individual to another by transferring immune cells نفسه from an immunized individual to unimmunized individual. So we have 2 types of active immunity and we have 2 types of passive immunity. Artificial active and natural active. Natural active when we are exposed naturally to the infection. Artificial active when we are using baskets. Now, passive immunity, natural passive, which when the mother gives the immunoglobulin to her baby through the umbilical cord. Artificial passive طب artificial passive when we take the immunoglobulin from outside the body from an animal or another human. طيب عندنا إشي اسمه the primary and secondary immune response. إيش يعني primary؟ primary يعني happens with the first contact of the antigen. With the first contact of the antigen. Produces what? Antibodies. It takes a longer period of time, and at the end, memory cells will develop, and naive B and T cells respond to the antigen. Now, the second response happens after a second or subsequent exposure to an antigen, like doses. تمام؟ يعني متقطع. Also, antibodies are produced, but in larger quantities, actually, and more memory cells will develop. طيب هون we have a patient have a hepatitis, which is inflammation of the liver, and we can see the hepatitis patient is given a Vascan and how it's given in 3 doses in order to reach a large and protective quantity of antibodies. Between the first dose and second dose, the are 2 to 4 weeks, and the third dose is given to 6 months after that. If you give fourth and fifth doses or an additional dose every week, for example, it will result in a shutdown of the immune system because the immune system will realize that there is no need for such immune reactions to take place. This technique is used dramatically treating allergy. It's called desensitization. After that, there is no need to repeat the process because there will be memory cells that will recognize the antigen. So it is a kind of response. هسا أنا إذا عندي مثلا hypersensitivity for a specific food, so I will start giving the patient a doses to make large and protective quantities of antibodies. بعطيه أول dose بعدين second dose بعدين third dose بس ما بعطيه أكثر من يعني ما بعطيه أشياء كثير. Why? Because it will shut down the immune system, because the immune system will realize that there's no need such immune reactions to take place. Okay, desensitization results in forming T regulatory cells. Well, T regulatory cells هم بيسموها suppressor T cells, they shut down the immune system. So these T regulatory cells or اسمهم suppressor T cells shut down the immune system. طيب هسا عندنا the independent versus the dependent response. And what do we mean by dependent and independent? Whether they need an MHC or MHC molecule or not. Now, the independent response, we have the B cells, and B cells binds directly to the antigens, and then it's activated. The dependent response, the antigen, the antigen is ingested, okay, and attached to an MHC molecule. This is the MHC molecule in the phagocytic cells. This is a phagocytic cell, and this is the MHC molecule, and this is the antigen. So the antigen is first of all, the MHC molecule is attached in the phagocytic cells, or APC. Then both will be displayed on the APC surface, the MHC and the antigen. هاي الانتيجين وهاي MHC. Then what will happen? The cell attached to this displayed complex, SOA complex, and becomes what? Activated. This T cells will attach to the complex and will be activated. What will happen next? It will secrete what? Cytokines, and this means cytokines. The cytokines will be secreted. Then what will happen? The cytokines which will activate the B cells and finally produce antibodies. وبس الله يعطيكم العافية، ألف عافية وبالتوفيق يا رب.